we can then increase the probability of having a baby as an outcome. Remember, in terms of evolution, right? Sexual reproduction's main purpose is reproduction, not fun. Right? So keep that in mind that when you don't get pregnant from having sex, the body will consider it a failure. So it's going to get ready for the next month. So then you can have babies the next month. So keep that in mind as we're going through this. So let's go through. I'm trying to find where I put my red one. So we're going to need two different colors, that's why. So it makes it easier when we see, right there, thank you. So let's talk about meiosis. Now, in meiosis, we're going to see, meiosis has one round of DNA replication, right at the very start. Then 2 rounds consecutive of division. So we're going to see, just like in mitosis. In mitosis, you have 1 round of DNA replication. Remember the cell life cycle, right? In the cell life cycle, it looks like a little pie chart. You see G1, S, G2, and then the phases. Prophase, metaphase, anaphase, telophase, and then cytokinesis. So that is the normal cell life cycle right here. In mitosis, you just go through one time complete. That's it. Now, in meiosis, you go through one cycle like this, and then you go back and resplit and redivide the newly formed cells. So then you can actually have more cells with 23 chromosomes. Because what happens is you have a stem cell right here. 46 chromosome, let's say it's an XY male stem cell, right? After the first round of cell division, now we have a stem cell that has 46 chromosomes, but they're now double armed right here. So when you see on this picture in meiosis, when you see a dual armed chromosome, right, we see here, that's a sign that that chromosome and that cell has undergone DNA replication. So it has undergone this S phase already. And what happens then? You have double the amount of DNA. Instead of having just single arm chromosome number 1, now we're going to have another arm to it. We call it chromosome 1 prime. Right? So what happens? You have one chromosome 1, and now we doubled it through DNA replication. That's true for all 23 pairs of chromosomes. So we have chromosome number 1 here, 1 prime, and then we have another chromosome number 1 that we inherited from our mom. So, right, here's the one that we inherit for dad. Here's the one we inherit from mom. So what happens is by the time we're done with the synthesis phase, we have doubled the DNA. Now here comes a problem. You have 46 chromosomes that you just doubled. That makes it 92 chromosomes. Kind of makes sense why you need to divide twice, right? If you divide one time, you just produce 2 cells with 46 chromosomes. Well, that's still too many. So each cell has to divide again. So that's what we see here. 1 round of cell division, and then immediately a second round of cell division. Now, the first round of cell divisions does a couple things right here. So all your chromosomes from dad will be in black right here. Here's chromosome number 2, here's chromosome number 2 prime, right? And then here's chromosome number 3, and then chromosome number 3 prime. And then here's dad's Y chromosome, being a little bit smaller, that we inherited from our parent, and Y prime. What we're going to see is that when we start aligning our chromosomes, alignment is incredibly important. And then X chromosome is actually pretty big compared to the Y. So, right, when we align in anaphase. No, in metaphase is going to be incredibly important. So here's my metaphase plate. In metaphase, in meiosis, right? Remember, metaphase means that all the chromosomes are found in the middle. M for metaphase, M for midline. What happens in metaphase is this. All of dad's chromosome number 1s, 2 copies. And all of mom's chromosome number 1s are going to line up side by side to form what we call a tetrad. Right? So all of chromosome number 1s from each parent will line up next to each other. Now, that's going to be important because when we separate these guys right here, as we separate them, the separation of those chromosomes are going to be what allows us to have 23 chromosomes each. So all the dad's chromosomes are lined up right here. Mom's chromosomes number 1s lined up right next to each other. Same thing with chromosome number 2 from dad. And chromosome number 2 for mom. So what's going to happen? When I divide this cell, or this half of the cell, we receive chromosome 1, 2 copies, but from one parent. This cell right here will get chromosome number 2, 2 copies, but from one parent. So what happens is, right, we don't have kind of a splitting where one of dad's chromosome 1s and one of mom's chromosome number 1s are kind of moving together. Here, all the chromosome number 1s that goes on one side will only come from one of our parents. Right, so chromosome number 3, and notice how I'm kind of purposely drawing this so then mom and dad's chromosomes aren't only on one side. There's a reason why, right? Well, we'll talk about in a second. And then the X chromosome from dad, or the Y chromosome from dad, and then the larger X from mom. Right? Now, what happens as well is this. Right? In metaphase, everything will align in the middle, and since everything is aligned in the middle, in anaphase and late prophase, what can happen is this. What can happen is some of dad's chromosome 1 can get switched, right? And because these chromosomes are so close together, there's a transfer of genetic material. So then a little bit of chromosome number 1 from mom crosses over and becomes part of dad's chromosome number 1. It's kind of cool, right? Now, it's kind of cool, except sometimes there's an unequal amount that gets shared. See how that happens? Right? So they're so close that they actually cross over, and there's a transfer of genetic material from some of mom's chromosome to dad's chromosome, and some from dad's chromosome to mom's chromosome. Why is this important? Well, now chromosome 1s right here, they were all identical, right? These two are identical. Now they're no longer identical. Right? So that gives us the ability to have a little bit of variation, even though we don't think we should. Now, chromosome 2 as well. That can also happen. There's another chromosome where this happens, and it really causes an issue. Chromosome number 21. Sometimes there's a little bit too much genetic material that's transferred into one chromosome, and you get what's called a trisomy 21. Trisomy 21 is Down syndrome, right? Where you just have either a little bit more chromosome 21 or you have a complete chromosome 21, right? That shouldn't be there. So instead of having only 2 copies, one from each parent, now you have a 3rd copy, trisomy. So that can happen where you have what we call unequal non-disjunction right here, right? One chromosome gets completely transferred almost to the other. Now, the good news is that doesn't happen as much as we think, right? And the other good news is usually when we're younger, it usually doesn't happen, and if it does happen, it's very minimal. As we grow older, that does happen though. Now, besides 21, where you can have trisomy, there's also trisomies in 13 and 18. Those are far worse than in 21. Usually those babies don't survive past infant years. All right, so we're in the metaphase plate. We talked about how there could be a transfer of genetic material from crossing over. And now we're going to have to split the first time. As we split the first time, we're going to align everything right down the middle. So all of chromosome 1 from mom will go to this side. All of chromosome 1 from dad, and a little bit from mom right here, goes to the other side. So as we split it apart right here, right, we're going to have the anaphase right here. And now in anaphase, here's dad's chromosome 1. Here's dad's copy with a little bit of mom's chromosome number 1 over here as well. Here's mom's chromosome 1 going to this side. And here's dad's chromosome 2. And here's mom's chromosome 2. Going to that side. Right? So what we have done is kind of split it in such a way that for each cell that we're going to produce, they're going to only have one copy from one, right? You're going to have two copies from one parent. So all the chromosome 1s right here, they'll turn into a different cell in the second, right? So in this cell, all the chromosome number 1s is only for mom. You have two copies, but they're both from mom. The chromosome 2, you have two copies, but only from one parent. Same thing here. Here's chromosome number 3, goes to this side. Again, two copies of chromosome, but from one parent only. Now, why am I making such an effort to mention this? The reason is simple, right? When we have two copies, they're attached, they're from one parent, right? We give it a weird number. We say you only have 23 chromosomes at this point. So this cell, even though it's got two copies, we say it only has 23 chromosomes. Why? Because they're attached to each other, right? And they share identical, supposedly identical genetic material. So it's kind of crazy, but that's why we say you have 23 chromosomes after the first split, and then you split again, but you still have 23 chromosomes, right? The reason why is that in reality, when you split the first time, you actually have 46 chromosomes. But because they're connected and because they're copies of each other, identical, hopefully copies of each other, they don't count as separate chromosomes yet. And then you have, again, the X chromosome. Actually, that's a Y. It just looks like an X. And that side, over here like this. Right? So the first time you divide, right here, you form two cells. Each cell is going to have one copy from one, it's like two copies from one of the parents right here, right? So 23 chromosomes, one through the sex chromosome right here, and now they're going to divide again. As they divide again, they're going to align very similar than what we see in mitosis. So here, all of the chromosomes then with the second division. So we'll take this cell right here, all right? We're going to pretend we divide it in half right here. We're going to take this cell, and now we're going to make it go through prophase and metaphase. So we just move it in metaphase. Prophase, you just see it, so it's not quite as important. And what we're going to do is align all the chromosomes on top of each other. Here's chromosome 1 from dad, the original. Here's a chromosome 1 from dad. That is a little bit of mom's. Here's chromosome 2. Here's chromosome 3. And here's our X chromosome, being very big. And they all are going to line up right in the middle, just like what we see in mitosis, right? So in this metaphase 2, all of the sister chromatids align in such a way that we're going to split it in half. As we split it in half, each cell is going to only have one copy of one chromosome, and that's it. So as we split this right here, this chromosome number 1 that's only from dad, that goes to this cell right here. This cell, right, on this side is going to get copy of chromosome number 2 and is purely mom's. This cell is going to have one copy of chromosome 3 going this way and is purely dad's. And then it's going to have the X chromosome for mom. So what we're going to see is, right, you're going to produce sperm cells, and those sperm cells that are produced are not going to be identical anymore, right? Because of a little bit of dad's genetic material and mom's kind of transferred. When we look at this, these sperm cells are going to be slightly different. So here's my sperm cell. On this side, this guy right here goes to the right. So this chromosome number 1 has a little bit of dad's chromosome and a little bit of mom's chromosome 1. On this side, this cell is going to have chromosome 2, a little bit of mom's chromosome 2, with a little bit of dad's chromosome 2. Again, why do we have this this kind of, switching and crossing over? Is to give us genetic variance. So then you can see that, you hear about some people saying like, oh, you look a little bit like dad, sometimes a little bit like grandpa, right? So that does happen because of this. Here's chromosome 3, and here's the other X chromosome, right? So by the time we're done, each of these cells is only going to have one copy of one chromosome, and that's it, right? That copy of the chromosome right there is going to be different than this cell's copy of the chromosome because of the crossing over event, right? Now, by the end of this whole thing, we're going to produce in sperm 4 sperm cells, each with 23 chromosomes. Perfect. Again, for males, the purpose of meiosis is to increase the number of sperm cells. So then you can increase the odds of pregnancy. Again, right, reproduction. We talk about sexual reproduction, right? We think about sex, the body thinks about reproduction. So the more sperm cells, the greater the probability that one of them is going to make it through and somehow going to fertilize that ovulated egg. All we need is one. But if you need that one, you want to have as many bullets there so then you can have that increased probability. Now, we're going to see in females, it's completely different. Do we want 4 eggs to be released each time we ovulate? That means you'll have 4 babies each time that you are going to get pregnant. That's going to be really tough, right? So what we're going to see is females, the meiosis is slightly different right here.